Thank you, Mauricio. Thank you, Rosella, for getting me to come here. And thank everyone here. Something else that I just found out about 2 weeks ago. How many of you have had a DNA swab to find out your human ancestral lineage? Some of you can pay like 70 euros and mail in.
National Geographic, right?
Yeah, you could. There's a few different companies that do it. And my family history comes from England and Ireland, but when I got my sample back, I was 5% Iberian. And so I have some ancestry here, so that makes me even more happy to be here. So I'm going to talk about a couple of things. Since I've never been here and I've never met most of you, I'm going to give an overview of work that my lab has been doing over the past decade or so. The picture in the background is of one of the sites that I work at in the White Mountains of New Hampshire, a site called the Bartlett Experimental Forest. A lot of the work that I'll show started there, and then we've kind of built out and scaled up to sites elsewhere in North America. And for those of you that haven't been to my part of the country, New Hampshire is in the upper right-hand corner, and the University of New Hampshire is a medium-sized university, 20,000 students maybe, about an hour north of Boston. It's a nice area. If anybody wanted to come visit, I would recommend it. Come in the summer, not in the winter, but it's a beautiful area. So right now, the three themes that my lab is working on are kind of shown in these cartoons here. One has to do with how ecosystems influence climate. We know that climate influences ecosystems, but ecosystems also have a strong effect on climate through their albedo and their evapotranspiration of water. So in New Hampshire, we have built several flux tower sites where we're looking at water, carbon, and energy exchange. I also have this project, which is primarily what I'm going to talk about today, looking at how we can scale the local measurements we make on carbon and nitrogen cycling up to larger landscapes and hopefully at some point the whole continent, or beyond that even if we can. And then a new project that, if I have time, I'll just touch on at the end, is looking at the effects of tree canopy biodiversity on carbon cycling. Most of what I've done so far is looking at climate and nitrogen and physical and biogeochemical factors. I was trained as an ecologist originally, and so I'm kind of trying to get back to my ecological roots by studying biodiversity and asking whether that plays a role in the carbon cycle and in how ecosystems influence climate. I'm going to start by going back 20 years, actually, which is hard to believe. When I was a graduate student, one of the projects I did was I took an ecosystem simulation model and some spatial data for vegetation from remote sensing and some climate data, and I tried to make predictions of forest productivity across the region that I'm from. And this, for those of you that don't recognize, this is the northeastern United States. New York City would be right about down here. Boston is here. This is the state of Maine. And Canada, Quebec, Montreal is about here. And so this goes back a long time when you couldn't download satellite imagery on your cell phone back then. We had to do a lot of work to create all of these climate data layers. And we ran this simulation model. And the model was able to generate hypotheses like that deciduous forests in our region should be more limited by precipitation. And so the patterns of growth you might expect would follow patterns of water availability, whereas in the evergreens, spruce, fir, and some of the pines that we have, they're not quite so water limited, and their growth rates, the spatial patterns that you see in this image within the evergreen forest would be regulated more by growing season length and growing degree days, more energy constraints or temperature constraints. So this was a starting point, and it wasn't bad. Some of these estimates are still pretty good, but I then followed up with a series of field studies where at some of the research sites that I work at, we went and measured forest productivity and looked at different factors, different environmental variables that growth rates were related to. These data are from 3 locations. 2 of them are in New Hampshire and 1 is in Massachusetts, which is the Harvard forest. And when we went and measured above ground net primary productivity at forested sites, at these research sites, there was a strong correlation with the concentration of nitrogen and foliage. which we didn't have any information about back then. Ideally, we'd like to have a map of nitrogen and foliage for that region I showed, so when we run our simulation model, we can include that as a variable that influences photosynthetic rates. At the time, there was a lot of interest in leaf area index. The MODIS satellite was being planned. And we had some ideas that maybe this relationship really stemmed from a relationship between nitrogen and leaf area index, but that didn't prove to be the case. Leaf area index and forest growth rates in this part of the world are not very, very well correlated, which was a bit of a surprise back then. So really we needed some way of getting at foliar nitrogen if we wanted to make some statements about patterns of productivity across a landscape. I was working in a lab where several people had been developing techniques using high spectral resolution remote sensing, which is, or imaging spectrometry, if that's a word that's more familiar. And it's a spectral instrument that takes the reflected sunlight off of the Earth's surface and breaks it up into something like 250 continuous wave bands that are very narrow, like 10 nanometers wide. So you can see very in detail the wavelengths of light that are absorbed or reflected by the vegetation. And we thought this was useful because partly what plants do for a living is they absorb some wavelengths, reflect others, and a lot of properties in the canopy show up as having spectral signatures in different parts of the reflectance spectra. So we got funded from NASA, which has this instrument. It's called AVIRIS, or the Airborne Visible and Infrared Imaging Spectrometer. It's flown on a high altitude aircraft, flies at about 20,000 meters. And for the location that we were interested in on the ground, when NASA was doing the overflights, we had field crews on the ground sampling lots and lots of foliage from many, many plots across the landscape. And after having field data from an ample number of plots, we use a variety of statistical techniques. The one that often works the best is partial least squared regression. And by doing this, you relate features in the reflectance spectra to the nitrogen concentrations that you measure on the ground. And if you get a good enough relationship, a good calibration relationship, you apply that to the imagery and you generate a map that shows predicted patterns of foliar nitrogen. This is an example of what one of these images looks like. This is the Hubbard Brook experimental forest. Some of the features that stand out, if any of you are familiar with the work that's been done at Hubbard Brook, it's famous for having done some clear-cutting experiments back in between the 1960s and the 1980s. And these watersheds, these bright areas up here, are small headwater stream watersheds that were experimentally clear-cut in the 1960s, 70s, and 80s. And because of the early successional tree species that are there and the stimulation of the nitrogen cycle that those disturbances produced, you see that in terms of very high nitrogen concentrations. On the opposite ridge, there's a lot of spruce and fir. There's some hemlock down on the stream channel in the middle. So the spatial patterns of this landscape follow what we expect when we think of the nitrogen concentrations of different tree species. We tested this method at a lot of different sites. These are predicted versus observed nitrogen values, including data from North Carolina, Florida, Massachusetts, a site in Australia. There's some eucalypt forest there. A tropical site in Costa Rica, and a number of other places that subsequently we went out and collected foliage and used this technique, and it works. We're not always entirely sure why it works, which is something that I'll get to in a minute, but it works relatively well when we have a closed canopy forest and an ample supply of field data. We also were doing a lot of other measurements on the nitrogen cycle. These are just some example results from this study looking at, for example, the lignin to nitrogen ratio in foliage was a good predictor of the carbon to nitrogen ratio in soil. And if you can remotely sense these things, then you can make an estimate of the carbon to nitrogen ratio in soil. This image here is for a region called the White Mountain National Forest. Hubbard Brook is down here, the place I just showed you. The Bartlett experimental forest is here. This is about an 80 kilometer wide landscape, roughly 80 by 80 kilometers. And we measured the mineralization rate in soil, The rate at which nitrate is produced in soil is related to the carbon to nitrogen ratio of the soil material that's decomposing. So we were starting to be able to get at patterns of carbon and nitrogen cycling, and we thought this was a useful tool for scaling across a landscape. There were several questions that came up, as is always the case. One of the things we were curious about is whether the relationships between canopy nitrogen and productivity and other ecosystem processes that we were seeing in the northeastern US, did they apply more broadly, or where would they break down, and what kind of ecosystems would those relationships no longer work? And if the relationships do mean something in other ecosystems, is there any hope for being able to estimate nitrogen in foliage for larger scales? The imaging spectroscopy is nice where you have an aircraft flight over your site, but it's very limited because it covers small pieces of ground. It's not something that can be done over large areas or continental or global scales. The Hesperi satellite that Mauricio mentioned is one that some people have been trying to get NASA to build and launch for years, and that would be a global hyperspectral satellite. So we really wanted to scale this up, both in terms of whether nitrogen was related to carbon assimilation and if we could say something about patterns of nitrogen more broadly. So the approach we took, this took about 10 years and 3 funding cycles. We didn't have plots that we could visit frequently like we do in our backyard in New Hampshire to measure growth rates. But in North America, as is the case in Europe also, there's a network of flux towers that are measuring the exchange of carbon dioxide between the ecosystem and the atmosphere. So we proposed to go to this forested Ameriflux sites and use the fluxes from the towers to look at the rates of carbon assimilation, and then use this approach of mapping the nitrogen concentration in foliage for the landscape surrounding the towers at a number of different locations. And these are examples of some foliar nitrogen maps for some of the sites that we worked at. There's the Wind River site in Washington State, Campbell River in Vancouver Island, a few sites up in Canada. There's one site I should have here, which Rosella worked at, which I'll get to in a minute, in Arizona, that we added later on. So we tried to cover the major forest types of North America, and it took, like I say, about 3 funding cycles, lots of kind of managing the aircraft overflights, managing field crews, and traveling to all these different sites. And I really had one graph in mind. It took 10 years to be able to make this graph, but this plot here is on the y-axis is the maximum photosynthetic capacity, or the maximum rate at which the whole ecosystem is able to assimilate carbon dioxide through photosynthesis. And this would be for the footprint of a flux tower, which is something like 1 square kilometer. It's maybe not quite that big, but it's a landscape. It's not a single tree or a single leaf. It's the rate at which the whole canopy is absorbing carbon dioxide. And down here is the nitrogen concentration on average for the footprint of that tower. And we use the aircraft imagery to estimate the mean nitrogen concentration for that 1 kilometer area surrounding the tower. And we do this for all of the different sites that we visit. And across the ones that are shown here, here's Wind River in Washington, an old growth evergreen forest, a site in Wisconsin, our Bartlett site in New Hampshire, Niwot Ridge is in Colorado, Indiana, North Carolina, Florida, so covering the major forest types in the US. And we see a strong linear positive relationship between the maximum rate of CO2 assimilation and the mean nitrogen concentration in that landscape. This upper panel is showing photosynthesis or carbon assimilation per gram of foliage, per unit foliar mass. And down here is the same thing, but it's the CO2 uptake per meter square of land area. Basically showing the same thing in 2 different ways. Interestingly, when we express this per, I'm sorry, I had that wrong. This is per land area, and this is per unit foliar mass. When we do this dashed line here is the relationship between leaf level nitrogen and leaf level photosynthetic capacity that comes from a global analysis done by Ian Wright et al. In 2004, the TRI data set of, I'm sure some of you are familiar with this. And so we looked at the relationship, the leaf level relationship for woody plants worldwide, and it matched pretty nicely in a way that we would expect the whole ecosystem level relationship that we were seeing with a very different method. We expect the whole canopy level relationship to be a little bit lower, to have a lower intercept because of self-shading in the canopy. Not all the foliage in a closed canopy forest is fully sunlit, whereas the leaf level data that went into that study were all sunlit, full light foliage. And then we were able to do other things like the nitrogen. Really what this was telling us was the light use efficiency of these ecosystems was strongly related to the nitrogen concentration of the foliage. This is a figure from a paper that Rosella published a few years ago when she was at New Hampshire. We were also interested in looking at whether or not some of these variables scaled with water use and water use efficiency. And so these are some of the same sites with the addition of that Arizona site, which is a drier site, looking at the relationship between nitrogen, water use efficiency, evapotranspiration, gross primary productivity, and rain use efficiency, which is similar to water use efficiency. So, you know, we think this is useful, and we think, for example, for models that need an estimate of photosynthetic capacity, if there were some way to estimate nitrogen concentrations over larger scales, it would be useful. If there isn't, then it's interesting. It's an interesting result, but it's difficult to apply over large areas. So the next question we asked is whether there was hope for estimating nitrogen over broader scales. And the answer is maybe, but something more interesting that we weren't expecting emerged from this data set. These are reflectance spectra for the forest sites that we visited. And we have data for all the sites we visited. We had field plots where we sampled foliage from about 180, 190 different plots. And a plot would be about 20 by 20 meters in area, which is the size of the resolution of the pixels that we were looking at. And so we were able to look at the field nitrogen concentrations and the reflectance spectra from the aircraft for all of those plots. I could show 180 spectra, but that would be messy. So what I've done here is I've taken the reflectance spectra and averaged them into nitrogen classes. So this black line down here is the mean reflectance for all of the sites that had foliar nitrogens of less than 1% by leaf weight, and then 1.1 to 1.2, and so on and so forth. And what emerged from this when we looked at this is that when you go from low nitrogen to high nitrogen canopies, the reflectance gets just sequentially brighter and brighter, not in narrow little bands like we had been looking at so far, but across pretty much the whole infrared region of the spectrum. You just get increasing brightness as you go from low to high nitrogen canopies, which is really interesting. It's not something that we expected, but it also shifted our thinking a little bit. I had not been thinking about albedo or surface energy fluxes at this point in this project. But if you think of the energy that's in sunlight, about 50% of the energy in sunlight is in the visible. These are the visible bands down here from the blue to the red. And about 50% of the just watts per meter square, about 50% of the energy in sunlight is in that infrared region. And when you get this big a shift in reflectance and how much sunlight is being absorbed by the Earth's surface or reflected, it starts looking like albedo, and it starts looking like something that might have an important effect on surface heating. And so we then started working with some people at Boston University who are developing the albedo product for MODIS. And we extracted the albedo estimate from the MODIS satellite for all of the sites that we had worked for the period in time that was as close as we could get to when we were on the ground doing our sampling. And across all these sites, we see this consistent, strong relationship between nitrogen and foliage and albedo in the canopy. And this, I hadn't really been thinking about albedo that much, but this is a substantial enough difference in albedo that if you have this kind of a shift over a large enough area, like the temperate forest biome of North America or Eurasia, it actually starts mattering in the context of surface heating in the way that climate models would care about. It doesn't take much of a change in albedo to really matter in terms of the Earth's energy budget. And it really was dominated by the near-infrared region. you know, this region here in the near infrared is really what's driving that relationship. There's not that much variability in the visible because these are all closed canopy forests, which are very good at absorbing visible light. And so regardless of the nitrogen concentrations and the photosynthetic capacities, the overall reflectivity in the visible didn't vary nearly as much as it does across the infrared. Just to be provocative, yeah.
See, in the previous slide, so the white point, no, the next one. The white point and the gray points, those are like conifers and angiosperms?
Actually, I should know, this is an independent data set collected by a different group. There was a paper from the Canadian Carbon Program. We wanted some independent data to see if there was something that, you know, we had done that was biasing this result. And so this was an independent data set. And subsequently, there have been other investigators who have looked for this and see a very similar pattern. I'll show some of that in a second. Just to be provocative, we took a midsummer albedo image from the MODIS satellite and we masked out all of the non-forested pixels of North America, and we used that relationship between albedo, nitrogen concentration, and photosynthetic capacity to basically suggest that at broad spatial scales, these three things kind of scale together. And that where you get very bright reflectances, you ought to have high photosynthetic capacities. This was really just a first stab at doing something at this scale, and it was meant to be provocative. We noticed that the region of highest albedo for North American forests is in the mid-Appalachian region, which is like West Virginia, for those of you that know that area. And I didn't know if that was real or if that's an artifact, if we were seeing reflectance off of rocks on ridgetops or something. But about a week after we created this image, I got a manuscript to review from the journal Ecosystems, which has subsequently been published, and it was called High Foliar and Soil Nitrogen Concentrations in Central Appalachian Forests. And it turns out that there are several research sites here, the Furneaux Experimental Forest in West Virginia, several that people at the University of Virginia have, and it turns out that area does have high foliar nitrogen concentrations, partly because of the species that are there, and partly, we think, because of the higher rates of nitrogen deposition that are there. Subsequently, there were a few other papers. The paper by Dave Ollinger et al., again using a different data set, saw a similar relationship between nitrogen and albedo. This data set, I don't have a legend here, but these are corn, grass, and soybean. So starting to look at ecosystems that are not forested. And then recently, last year, a paper out of Chris Field and Joe Berry's lab at Stanford was looking at global patterns in solar-induced fluorescence from the GOME satellite and gross primary productivity from a Max Planck data product. And they found the same near-infrared, the same region that we were finding is so strongly related to nitrogen, they found was the best predictor of solar-induced fluorescence and gross primary productivity. And so we've been talking with them more recently, trying to exchange ideas about why that relationship might be there. So it brings up the question, you know, we thought we needed this narrowband imaging spectroscopy data to estimate foliar nitrogen concentrations, and is that really important? For all of the field sites that we have collected data from so far, the best prediction we get from the narrowband imaging spectrometer data is this is the predicted versus observed value here. And if we simply use one big fat NIR band, like a 50 to 100 nanometer wide band centered right on the near-infrared plateau, statistically we get a strong correlation between NIR reflectance and the foliar nitrogen values that we've measured on the ground. And does that mean that there's existing vegetation indices that are strongly driven by the near-infrared band? Does that mean that things like NDVI or EVI or the ratio vegetation index should be related to foliar nitrogen also? And the answer is no, for interesting reasons. We've looked at the relationship between foliar nitrogen and all these different vegetation indices at different spectral resolutions. So taking our imaging spectrometer data, we can calculate NDVI at 10 nanometers or 20 nanometers or wider bands, and we never really see a strong correlation between those vegetation indices. But regardless of the bandwidth, we do see a strong relationship with the NIR. And here's why. if we look at individual relationships between individual bands, these are bands selected to mimic what we have on the Landsat satellite, which has a blue band and a green band and a red and a near infrared, all compared with our field measured foliar nitrogens for the locations that we've been working. No relationship in the blue, green, red, and as soon as you hit the NIR, there's this strong linear correlation. And then it goes away as you go into the longer wavelengths. So by calculating an NDVI type vegetation index, effectively what you're doing is taking this relationship and adding noise to it. So we don't see a strong relationship with NDVI. We really just have to look at a single reflectance channel in the near infrared. So by implication, there should be some instruments, if the signal to noise ratio is high enough and the data quality are good enough, which is an important caveat, we should be able to start looking at patterns with things like Landsat or perhaps the Sentinel instruments. We've tried this with earlier Landsat instruments, but the data quality just are not high enough, and it turns out the signal to noise ratio is too low. This is an image for the Harvard forest in central Massachusetts. This is these polygons are the areas that make up the Harvard forest. And this is a superimposition of an Avarice-based nitrogen estimate versus one that we created using a single NIR band from Landsat 8. Landsat 8 was launched in 2013. We don't have cloud-free imagery for all of the northeastern US yet, but I think it's at least promising. But it begs the question, why would nitrogen concentrations in foliage and near-infrared reflectance be so strongly correlated? Nitrogen itself has no distinct spectral properties. It's in foliage, most of it is tied up in rubisco, the carboxylating enzyme in photosynthesis, and as well as pigments, chlorophyll and other pigments. Pigments are active in the visible part of the spectrum, not the near-infrared part of the spectrum. But from what we can tell, there really is no clear mechanism through which nitrogen should be influencing near-infrared reflectance. So it must be some kind of covariation between the nitrogen concentration through the effect that it has on photosynthesis, how that then influences carbon allocation and optimal patterns of light interception by foliage, and other things that we haven't fully put together yet. We've tried to investigate this using some modeling and some field measurements. These are modeled or predicted leaf level reflectances from a radiative transfer model called Prospect. And you can do things with the model like hold all variables constant and look at the effect of varying chlorophyll. Chlorophyll is active in the visible, so as the chlorophyll concentration varies, you get a lot of difference here, but in theory, not really a difference in the infrared. Leaf mass per unit area has an effect on the near-infrared plateau. It could be something to do with leaf density or leaf thickness. There's a cell structure parameter in Prospect that appears to be very sensitive. The model is very sensitive to that. Possibly leaf water as well. But we don't think so. Leaf water, the water content of foliage starts absorbing kind of roughly, at least heavily. There's several absorption features in the near infrared, but really when you get out beyond 1400 nanometers is where water has the biggest effect on reflectance. You can also use a canopy model that takes that leaf level model and couples it with a full canopy, and you can manipulate things like leaf area index or the leaf inclination angle, whether leaves are upwardly oriented or whether they're oriented more horizontally. And these things also look like they potentially could have this effect. So any one of these things that is in some way mechanistically tied to nitrogen could result in that relationship that we see. There are other radiative transfer models that make different assumptions about what influences canopy reflectance. Things like crown shape and crown depth, which the ProSail model doesn't have in it, leaf size and shape, stem architecture. And they all make what they're trying to deal with is the fact that if a photon is coming from the sun and it starts interacting with foliage in a canopy, if the first foliage that sunlight interacts with is in the upper part of the canopy and it is reflected back to space, if there's a sensor there, the sensor will see it. But as photons are able to penetrate more and more deeply into the canopy, if they're reflected by foliage down in the mid or lower part of the canopy, they stand an increasing probability of being absorbed by the underside of another leaf. And so the vertical structure and the vertical distribution of foliage in the canopy, or the vertical distribution of where sunlight is starting to interact with foliage, theoretically should be what's driving that near-infrared signal that we see. Interestingly, if you think about the leaf traits, the stem level traits, and the canopy level traits that influence near-infrared reflectance, a lot of them end up co-varying across species because of a functional convergence, I'll call it, between the optimal growth strategies of different types of plants. An example, this is a spruce tree. A spruce tree has a in general, very low foliar nitrogen concentration, adapted to growing in poor conditions, cold climates or poor soils. So because of the type of life history that spruce has, it's got a low photosynthetic rate. It has to last a long time. The foliage in spruce has a leaf retention time of like 5 to 10 years on average, which results in a need for the cells in spruce needles to be very densely packed and durable, so they can make it through multiple winters. That has the effect of making leaf level reflectance dark in the near infrared. Because it also has a low photosynthetic rate, at the end of the growing season, there's less mobile carbohydrate available for stem elongation or lateral branching. So you also end up with needles that are tightly clustered on a stem, and you end up with a whole canopy that has a kind of a denser structure. And all of those things at the leaf, the stem, and the canopy level tend to have this effect of increasing the interception and absorption of photons and increasing the depth in the canopy to which photons can penetrate. And as you go to other species that are in our region, eastern hemlock, white pine, oak, all of these things that influence reflectance at the leaf level, the cellular level, the branch level, and the crown level all change in a predictable way because of the effect that photosynthetic capacity has on how much carbon is being assimilated, and then how it's allocated in the canopy, and how that leads to differences in leaf display. That's all kind of vague and nebulous in a way, and I wish I knew exactly which of these different parameters mattered the most. But we're convinced at this point that there is a mechanistic underpinning to this link between nitrogen and reflectance. And something that I just think is interesting is to ponder is you end up, in terms of looking at photosynthetic capacity, we end up learning more about the functioning of plants by examining the wavelengths of light that plants don't use, which is the near infrared, than by looking at the wavelengths that are, at least at this scale and in this context, which I think is interesting. It may be an accidental consequence of some of these relationships, or it may be something about plants with a high photosynthetic capacity that maybe are more susceptible to heat stress and have a greater incentive to dump near-infrared light. That's basically the waste product wavelengths, the wavelengths that they're not using. And finally, I'll say a little bit about this other project that I've been working on, which is trying to get away from just thinking about climate and nitrogen and thinking about other ecological factors. And this is a new project thinking about canopy diversity and whether there's some influence of the diversity of species in a canopy that also has an effect on carbon assimilation. And this is review for most of you probably, but why would diversity influence carbon assimilation or other ecosystem processes? And there really are two primary reasons. One would be the resource acquisition hypothesis, where different species have different strategies for acquiring resources, and there are trade-offs between the different approaches that different species make. Some are more efficient at low resource supply, and some are more efficient at high resource supply. And if you have multiple species present, in theory, you get more complete or more efficient exploitation of resources in a multi-species ecosystem. Then there's the diversity stability hypothesis, which says that during times of stress, if there's a drought stress or some other stress, if you have many species present, you're statistically likely to have at least some species present that can tolerate that stress and effectively pick up the slack for the other species that might not do so well. And this hypothesis was probably, it's been demonstrated a number of times, but the first and clearest demonstration was the biodiversity manipulation experiment at Cedar Creek in Minnesota by Dave Tillman. This is a short grass prairie, a grassland where they artificially manipulated the number of grass species in a number of different plots. And after they set up this experiment, there was a severe drought that came in, and the response, the degree to which growth rates declined during this drought was related to how many species were there. Where very low diverse sites had a much greater decline in productivity than more diverse sites. But there's one issue, is most of what we know about this influence of diversity on ecosystem function comes from either grasslands or other small statured ecosystems where you can manipulate the plants, where you can clear a site and plant multiple combinations of different species. It's difficult to do that in forests, mature forests, because of the size and longevity of trees. It would take you 50 to 100 years after setting up an experiment to be able to study this. So we've been taking another approach, making use of the data that we've collected as part of this project, looking at the data from the Ameriflux network and looking at the differences in tree diversity across this gradient and trying to see if we can identify an influence of diversity after accounting for the effects of climate and soil type and all the other factors that vary at this scale. And the weakness of doing this kind of large gradient analysis is that there are all these other things that co-vary with diversity. We took this approach because the FluxNet data probably have about as dense a network of other meteorological and site level variables. So if we have any hope of being able to identify a diversity effect on things like gross primary productivity after accounting for climate, soil, vapor pressure deficit, and other factors, we think this is at least one way to try. So we filtered out the data that we've collected so far, the data that are available from these sites. We limit the analysis to forested sites that are at least 10 years old. They have a full canopy. There's enough vegetation data to be able to calculate a diversity index like Shannon's diversity index, which is one common one, and have lots of other site level data, the micrometeorology, the drought severity index information, foliar nitrogen from the work that we've done, biomass, stand age, and the idea is to take all this information and build a statistical model that looks at factors that explain variation in evapotranspiration and gross primary productivity across this region, and see if diversity still stands out as having any kind of an interesting or positive effect. And very briefly, in the analysis we've done thus far has used monthly gross primary productivity and monthly evapotranspiration data. It turns out you can predict monthly GPP with these variables with an R-square of a little over 0.7. As expected, PAR, air temperature, vapor pressure deficit, Palmer drought severity index, foliar nitrogen, those are the factors that have the biggest effect on patterns of gross primary productivity. But no matter how we bracket this data set and no matter how we run this statistical analysis, the diversity index consistently comes out as having a positive effect on gross primary productivity. And if you look at the slope of the effect in that model, it's equivalent to something like 6 grams of carbon per meter squared per month for each additional tree species that you have present on a site. It's not huge, but it's significant. And it might be a way of getting at whether this is enough of an effect as we go beyond looking at just monthly fluxes and we can do this from daily to annual timescales if an effect of that magnitude consistently stands out. And then for sites that have enough years of data, we can look at the interannual variability in the fluxes. And in theory, if that diversity stability hypothesis is borne out, you should have less interannual variability at more diverse sites. And we see that for gross primary productivity. We don't see that for evapotranspiration. It's unclear why, but thus far this is where we're at with that project. So we have some support for both of those hypotheses in forests. And the next step is to go out and use LiDAR and other types of remote sensing to get at other metrics of diversity beyond the species richness or Shannon's diversity, but to look at structural diversity, spectral diversity, and a number of other factors. I think I'll leave it there. I have a little bit more, but that's probably a good place to stop. So I will say thank you. These trees here in this image are American chestnut. One of the reasons for studying diversity is even though temperate forests in North America don't stand out as a biodiversity hotspot, but because of invasive pests, pathogens, invasive insects, the number of species and the diversity of forests across the continent really is kind of slowly getting ground down. This used to be one of the dominant deciduous tree species in eastern North America, but because of a fungus that was introduced in the early 1900s, this one has been eradicated. And we're in the process of losing a number of others due to invasive insects and a few other pathogens. Anyway, I'll stop there and would be glad to take questions.
Many thanks. That's excellent talk. Really stimulating, lots of interesting points to discuss with the public.
Yeah, Scott, you know that everybody asks you about that, but they still, it's not clear, as you said, what really drives this relationship between nitrogen and the reflectance. Wouldn't you say that all these variables that they scale together, at the end you reach this kind of conclusion? It's a set of several traits that only scale up the same way. At the end, what you are seeing there, isn't that just biomass structure?
It's not. Biomass or leaf area index don't show that same correlation. They do if you look at systems that have leaf area indexes of like 2 or lower. But once you get to an LAI of 3 or higher, it's a light saturated closed canopy system. And foliar biomass and leaf area index don't show that same relationship.
This is the same thing that happens with the new product by Joe Berry,
right.
So,
yeah.
I don't see it clear, but
yeah.
If you are sure of that, because what you see is that at the end it's not only biomass, but it's structure to that.
Right.
But the structure is related to biomass.
Right, right. And it is structure. So when I talk about things like leaf angle distribution is part of structure. You know, you can have 2 forests that have an LAI of 4, and depending on the, so here's a hypothesis that I didn't articulate that well. If a leaf has a high nitrogen concentration, it has the ability to make more efficient use of full sun. And so if you run a leaf level optimization model, you'll predict that leaves with a high photosynthetic capacity should be oriented more, with more direct surface area facing the sun. And as you get to leaves that, like eucalypt forests, we went to Australia to study eucalypts. They're notorious for being adapted to poor soils, and they often have a very vertical leaf inclination angle. You can have a eucalypt canopy with an LAI of 5 or higher, but because of that vertical leaf angle distribution, the photons that come in are able to pass down into the lower parts of the canopy more easily than they would in a canopy where leaf angle is more upwardly oriented. And that has the effect of trapping photons down in the canopy. If they're reflected off of something, they don't as easily make it back to space. And so the near-infrared reflectance in a eucalypt forest, because of that structure, it's not leaf area index, but it's something, it has to do with how the foliage is displayed relative to optimally absorbing sunlight. I can't explain it better than that. That's the idea, that's the hypothesis that we're working with right now. Ideally, what we would like to do is measure light response curves in plant species at different light levels and different nitrogen concentrations at all these sites to see if that is the explanation. That's what Joe Berry thinks it is. So,
have you tried also to still work with the traditional red edge and all these other ways?
I'm sorry?
All these other reflectance indices and reflectance algorithms?
We have, yeah, and believe it or not, a single near-infrared band always gives us a clearer relationship, and other people have seen it as well. At this point, I'm at least, it's occurred independently in so many different data sets, it can't be an accident. You know, things like that don't appear in nature by accident.
Well, no, the other thing is that the scale, so this convergence that you mentioned.
You're right, right. But it is important, before we can really use it and before we just can run wild mapping nitrogen with near infrared, we need to know what it is that's driving it, because you know, let's say it is a structural effect of the canopy. Well, if you have selective harvesting and suddenly because of logging there are trees that have been removed from above, you'll start having these dark shadowy spots, and you know, that will lead you to a conclusion about what the nitrogen concentrations are at those sites. That would be misleading if you didn't know that. If you're going to try to use this in a model, you'd also want to know what the mechanism is. If it's something like leaf angle distribution, plants can, that's a response that can happen fairly quickly. You can fertilize a site and water and irrigate a site, and you'll see a change in leaf angle distribution relatively quickly. But if it's crown architecture, if it's lateral branching, that's not going to happen overnight. That might never happen. You know, if the trees are competing with each other for sunlight, then you might never get an effect on crown architecture. So I'm convinced that there is a mechanism there, but until we really understand what it is, we have to be careful with, you know, using it to go off and map things. But at the very least, it's interesting, and it's just, to me, it's interesting that there's a link between nitrogen, photosynthetic capacity, and albedo.
So you didn't mention water. Sorry, I didn't. You are
Okay, thanks. Again, on the same topic, on this relationship between nitrogen and reflectance, do you see it as clearly when you use nitrogen per unit mass, per unit area, sorry, instead of using it per unit mass? Because I guess that could give you an indication of what the mechanism is behind it.
Right. I didn't show this, but we've also looked at this relationship with leaf level reflectance. You know, we have a portable spectrometer, and you know, we can look at the nitrogen levels per unit area and per unit mass. Interestingly, and this is another thing that tells us that it has to do with canopy structure, at the leaf level we don't see that relationship. We only see it when we look at the whole canopy. And it's there whether we express nitrogen per unit leaf area, or per unit area, or per unit mass.
What about per unit of ground area? Leave nitrogen per plant surface?
No, that we don't, because you can have situations. So the Wind River forest site in Washington State it's an old growth Douglas fir forest. The trees there are you know, 90 meters tall, really tall trees. Leaf area index of 8 to 9, huge canopy biomass. These are really big old trees. And the total nitrogen content would be very high because of how much foliar mass is there, but the concentration in leaves is low. And in the near infrared, it's very dark. So that's an example of a site where it follows the pattern you expect based on concentration, but not the total content of the canopy. And the same is true with gross primary productivity, because given that if there's a leaf area index of something like 8, the more layers of foliage you add, those additional layers are going to have lower and lower levels of light. And it turns out that for total gross primary productivity, it's really the upper several layers of foliage that have ample sunlight that are driving the variability in GPP.
I guess one question I have is about water. You briefly mentioned the irrigation example before, and also the issue that below leaf area index of 2, then the relationship begins to...
Right.
And so that's, you know, leaf area index below 2 is really when you begin to see the drought stress.
Right.
And so what happens then?
What happens then is that this stops working as well. And in some cases, I think the link between photosynthetic capacity, nitrogen, and water probably makes this relationship, you know, vary through time in ways that I don't fully understand. You know, when leaves dry out, structural things happen to them. Sure. They can droop. You know, that's one of the responses to drought, is that leaves will droop. And as if you get a response like that that affects the structure of foliage or the way that the foliage is displayed, you'll see it in the near infrared in ways that are being driven by water, not nitrogen.
But you would expect the Mediterranean vegetation or drought desert vegetation to be adapted to that, you know, and so not really being below the loss point anyway.
Right.
They should be able to cope with.
Right. Right.
In fact, the turgor water content in leaves is very conserved.
Right.
Generally leaves don't really want to become very, very dry.
Right.
So I'm not quite sure how much of a difference that would make, but
Yeah.
it does suggest to me that well, it makes me wonder whether there are still structural properties at a broader scale where you incorporate a larger range of
Right.
different indices.
You know, and I don't know the answer to that. One of the reasons I'm traveling in Europe and visiting this site is to try to like talk to people who are more familiar with different kinds of ecosystems. My guess is that the structural properties of the kinds of systems you're talking about drive reflectance, but that they might not be related to nitrogen levels the way that they are in these, you know,
of course,
more.
the structural relief adapted to drought.
Right.
It's very different, you know, in terms of walls and
Right.
other compounds, cuticles, etc. To probably makes reflectance properties very different.
Right. Right. Yeah, I wish I knew the answer, but I don't.
Go ahead.
Just a little bit. By the way, the three subjects you are working on, we are very interested, Scott, you know. And the other one that we are very interested is also in these biophysical effects. Yeah. For example, one could be surprised to see what we teach in our classes. It's very simple, is that the albedo decreases with afforestation. And afforestation, instead, you show,
Right.
You show increases in albedo with increasing biomass and increasing.
Right.
We must be careful explaining this ourselves. So this is the point. But the other thing we work and we are really interested is also in diversity effects on stability and production. Yeah, we have similar projects. We will talk later on that. And there was a result that I was surprised. Maybe I am mistaken, but you had that biomass didn't affect GPP in your models.
That's right.
And isn't that surprising, or am I missing something?
Yeah, it is surprising, but yeah, it just did not come out as one of the factors that was a significant predictor of GPP.
Maybe because the range is not a large range.
I think partly, you know, because of the age range, some of the sites are young enough where the photosynthetic capacities of young plants are going to be very high, and there's not a lot of biomass there. There's not a lot of woody biomass, but there's a lot of foliar biomass, and that's what's driving GPP more than woody biomass. But that's a guess. But.
Okay, any more questions? Well, then we thank again Scott for the very interesting talk and wish you all the best for future work.
Yeah, thank you all.